Thank you for your introduction. Hello, everyone. Welcome to today's webinar session. In this session, I will explore and show you a brief overview of the challenges faced by cell and ex vivo gene therapy developer and how the GenScript can support you in streamline your process development and commercialization activity. Here is an overview of what we will cover in this webinar. We will start with an introduction to our company, and following that we'll share overview of the key challenges, and the next we'll introduce the key technique and service or reagent needed for each stage of the cell therapy development and their importance. At the same times, I will also uh, share how the GenScript strength and capability can accelerate the process of bringing your cell and gene therapy to the market. Uh, so, GenScript is a New Jersey based life science CRO with global presence. It's offering the wide range of service, uh, mm, products, and instrument to the pharmaceutical and biotech company worldwide for drug discovery and development. And GenScript ProBio is a subsidiary company which is providing end-to-end solution. Mm, and they are the CDMO service provider for drug discovery to commercialization in CGT field or vaccine or biologics drug discovery development. Another one is the Legend Biotech, which is another subsidiary company of the GenScript, which established in 2019 and 2021. Legend CAR T cell therapy received US FDA approval. And the last is the Best Enzyme, which is also the subsidiary company of GenScript, which is a specialized in synthetic biology product, and they providing end-to-end solution for the different kind of enzymes and the small molecule, etc. And in summary, GenScript has a long-standing record uh, of innovation and excellence in the life science, and it is well known as one of the top provider of the service and reagent, facilitating the advancement of cell and gene therapy from laboratory to the clinical setting. Uh, GenScript headquarters are located in Piscataway, New Jersey, with additional research and manufacturing site at the Seattle. Along with that, we have also the facility in Singapore and China for the manufacturing and the production. And we have regional office in the Europe, Korea, and Japan. As a result, we empower the researcher and clinician and biotech companies globally with cutting edge technologies and service, and ultimately translating the scientific discovery into benefit for the society. So our mission is to provide innovative technology and solution worldwide that boost advancement in the life science research, biological development, and cellular therapy. And ultimately, our goal is to enhance human health and well-being. GenScript has made significant contribution to the scientific community by facilitating the publication of over 1,000 K research paper in peer-reviewed journal. And these publications are demonstrate the impact of our products and services on advancing the scientific knowledge and how we support the researcher worldwide. So, after introducing the GenScript, their mission, and their values, in this section, I will provide an overview of the cell therapy and discuss common challenges generally faced in CGT field. The standard manufacturing process of CAR T cell therapy include the cell isolation, that is the first step. But the cell isolation also depend. The starting sample may be coming from patient if it's autologous cell therapy, and it's coming from the healthy donor if it is allogenic cell therapy. And after the cell isolation, they undergo cell activation, cell engineering, and cell expansion. And throughout these steps, various quality control measures, safety assessment, and regulatory compliance checks are performed to ensure the safety and efficacy of the cell therapy. So, the cell engineering steps involved in the cell therapy depend on the type of cell therapy being pursued. For example, when working with the TIL, which is a tumor infiltrating lymphocyte, the process involves extracting the tumor cell from the tumor, expand them into the laboratory before introducing them into patient. So in this TIL cell therapy, generally cell engineering is not needed. While working with the TCR or CAR TCR cell therapy required genetic modification of the patient T cell to express the receptor that can recognize and attack the tumor cell, and hence require genetically engineered cell for the therapy. So this is the common workflow, and in each step they need different kind of tools and technology. So as we mentioned, like cell therapy manufacturing is a complex process and involve manual and automated step. In addition, researchers are trying to improve this cell therapy manufacturing process to make it more effective, stable, and reproducible, while also aiming to increase the product yield, batch to batch size, consistency, while reducing time and cost. So, cell therapy manufacture involve multiple products, including ancillary and raw material, and the utilization of various raw material from different sources in a high complex process significantly impact the safety, reproducibility, cost of good, scale up and scale out capability, and quality. In addition, in addition, researchers are also trying to improve the sorry. In addition, any disturbance in the supply chain for essential product or consumable can cause significant delay in the production of life-saving drug. Not only that, finding the new product which can work well with the existing manufacturing process and optimizing the use of the replacement product can take months. So, to prevent the GMP complication, choosing the right service provider who can provide specialized reagent regulatory uh, with the regulation and regularly over a long period with the desired quality on time is very crucial. To address this challenge, GenScript provide top quality research grade and GMP grade reagents and service with supply chain guarantee. And we offer the phase appropriate reagent customizable solution. Along with that, we also provide the regulatory compliance support and robust automation capability to advance your therapeutic development. So, in this section, we will explore how GenScript can effectively aid in the mm, critical steps of the cell therapy process: isolation, activation, expansion, and cell engineering. I will walk through tools needed for each stage and their importance and GenScript's strength and capability. So first, which is cell isolation. For that, GenScript has to develop the cytosine technology to support the cell therapy field, and this technology is particularly helpful in ex vivo cell therapy, where the isolation of desired cell or depletion of unwanted cell is necessary. To facilitate this, GenScript offer antibody couple magnetic bead. All the key ingredient used for developing this bead is fully produced in-house, including the antibodies and magnetic bead. And these ingredients have undergone vigorous validation within R&D and with beta tester to ensure their quality and effectiveness. To support the reliable and efficient solution for isolation process, GenScript cytosine technology offer a comprehensive toolkit which consisting antigen-specific paramagnetic nanobead along with the automation equipment and the tubing sets. And these reagents are available for various marker like CD4, CD3, TCR alpha beta, CD34, CD56, CD14, and we also providing the SAB nanobeads. So customer can use antibiotic antibody and use the SAB system. And these reagents are sterilized, non toxic, available in both RU and GMP grade, and they are suitable for use both with fresh or frozen PBMC or leukophoresis, and capable to do both depletion as well as enrichment. And this nanobead do not interfering in the downstream application, and it's not interfering even in flow cytometry analysis, and working very well with the translational research. So for cell isolation, GenScript also offer the cytosint 1000 equipment, which is a automation equipment coming with the consumable tubing set, and this is highly efficient and automated equipment, and it's easy to operate and equip with the key features like the barcode scanning, and it's customizable programming, real-time workflow monitoring, and data integrating with the full traceability. Moreover, it's efficiently integrate with other instrument and work to in the modular system, and facilitating the smooth workflow for both preclinical and clinical development. So another one is the after cell isolation. In the T cell therapy workflow or NK cell therapy workflow, the cells go undergo the activation. So here in this experiment, T cell were initially enriched using the cytosine nanobead, and were activated using the GenScript and seed T cell activation reagent, and which was compared to competitor product also. So, achieving the less differentiated T cells, such as the naive or stem cell memory T cell during the activation step, is crucial. And if we see here, GenScript and T cell activation reagent enable the high-fold expansion while maintaining the stable memory phenotype, ensuring the enhanced downstream application and preserving the high cell viability. Not only that, but the GenScript cytosine activation and isolation bead have been registered with the Drug Master file on with the FDA, and to advance the development of cell therapy industry and expedite the process of cell isolation. GenScript entered into strategic collaboration with PersonGen, and this collaboration aimed to accelerate the progress of cell therapy, ultimately benefiting the larger number of patients. GenScript also entered into strategic collaboration with the Applied Cell to deliver the combined cell isolation solution for cell therapy drug development worldwide. Under this collaboration, GenScript will develop and supply its proprietary research and GMP grade cytosine reagent to used in the cell mars platform to developing the cell therapy workflow. So this is the applied cell mars equipment, which also work well with the cytosine nanobid. After the cell isolation and activation, uh, and after the cell engineering step, generally cell recombinant protein, cytokines, and growth factor are necessary ancillary material utilized during the cell therapy process. Example, like in CAR T cell therapy, T cell required growth factor to expand, which are provided by the serum. And one of the growth factor is IL-2, which upon binding to T cell receptor promote the polyclonal expansion of the T cell. And for that purpose, GenScript offer various kind of cytokines and growth factor, which can help to different type of cell proliferation and differentiation. And even if these recombinant proteins are not present in the final product, their quality can impact the stability, safety, potency, and purity of the product. So, GenScript providing the GMP platform to convert the RUO grade protein to GMP grade, and they are suitable for use in preclinical and clinical stage. And this can help in smooth transition of your project from research to GMP. So after the getting the information about the isolation step, activation, and expansion, the very unique step in the cell therapy is a cell engineering. So here the blueprint of the car design shown, and generally the car have the four main part, and which one of them is antigen binding domain, a hing region, transmembrane domain, and intracellular signaling domain. And each of these parts serve as a specific purpose, and the car can be designed optimally by using different combination of those protein domain. To enhance the efficacy and precision and safety of therapeutic cell, a variety of tools and reagents are essential to carry out this CAR-T optimization, means engine to optimize the engineering step. And that can be carried out by either viral using the viral vector as a delivery or non-viral method to achieve the best car design. So in this section, I will share the GenScript capability to support both the viral and non-viral way to do cell engineering. So in non-viral, we are providing CRISPR component, mRNA, circular mRNA, neoantigen peptide. And in the viral vector also, we are providing viral vector and plasmid as a vector, and both the types of vector AAV and LVV. So I will walk through each of them and show what the GenScript has and how it's work, and that will give you to select which one best fit in your workflow. So many methods for genome editing have been emerged, but one stands out is a CRISPR. It's gaining popularity as groundbreaking gene editing technology that empowers scientists to make precise alternation to the DNA, and these cells can be edited inside the body in vivo or outside the body ex vivo from patient or donor to the donor cell. Ex vivo CRISPR genome editing is a therapeutic approach in which genome of cells are edited in vitro, and then those modified cells such as T cell or HSC are transplanted back to the patient to produce the therapeutic mm, and resulting produce the therapeutic cell due to the genome editing. And when it comes to delivery of CRISPR and Cas9, there are basically two routes. One is non-viral transfection and viral vector transduction. And researcher can select which delivery method and types of reagent will going to work for them. So up to but we have like complete solution in whatever the way you are working. We have different kind of tools and material and service are available to carry out the cell therapy product manufacturing commercialization activity on time and with using the different tools. So currently using CRISPR's number of clinical trials are under investigation for different diseases, including the blood disorder, cancer, diabetic, infection disease, or inflammatory disease. And I will show you how GenScript can support your CRISPR-based cell therapy development work. So the CRISPR-Cas9 system involve the target-specific CRISPR gRNA and then Cas9 nuclease, and it's also involved the HDR template or DNA payload. We offer all these components in multiple format to give you flexibility in experimental design. Furthermore, we offer the Cas9 SGRN HDR template in from research grade to the GMP grade. So in upcoming few slides, I will discuss the various option of Cas proteins offered by the GenScript. So, through our partnership with Fangzheng Laboratory at the Broad Institute of MIT and Harvard, GenScript offer validated Cas9, Cas12, Cas13 nuclease product, and with CRISPR genome editing involved, application moving from basic research to the clinical manufacturing, high quality raw material and complete documentation are becoming more essential. So fulfill this in increasing specific increasingly demand and very strict specification. GenScript offer GMP grade gene CRISPR Ultra SPK9 and Ultra ESPK9 NLS. Both are manufactured under the GMP guideline, including aseptic manufacturing and documentation and QC testing. And GMP grade Ultra SPK9 and ESPK9 nuclease deliver high knockout and knock-in efficiency, which has been demonstrated both by testing in cell line and the primary T cell. And GenScript can assist with you clinical filing by offering documentation, even allow the conducting the site audits and providing additional support tailored to your project need in specific region. So they are come with the high editing efficiency, lot to lot consistency, ultra high stability, and with the stringent specification, means very strict QC criteria we set up to make sure that we deliver the good quality protein for the genome editing work. Guided RNA are the foundation of any CRISPR based gene editing experiment. Therefore, the design of gRNA is a critical step and ensuring because that will affect the successful gene editing or cell engineering outcome. So, to maximize this efficiency of edit and avoid off target effect, it's crucial to be careful while selecting the sgRNA. And in upcoming few slides, I will discuss various options of sgRNA we delivered and their advantages and their performance. So sgRNA can be synthesized chemically, which is known as a chemically synthesized sgRNA, or it can be synthesized in vitro, which is known as IVT sgRNA. IVT method for sgRNA generation suffer from limitation such as variable editing efficiency, high off-target effect, high cytotoxicity, poor stability, low editing efficiency, non-guaranteed success rate, and large difference among the batches. To overcome this limitation of IVT sgRNA and support the commercial production of synthetic sgRNA, GenScript produced the high quality sgRNA through chemical synthesis. Using the semiconductor microarray technology, GenScript accurately synthesize and verify sgRNA sequence are available for that work. And this cutting edge sgRNA synthesis method is not only more efficient, safe, and less toxic, but it's than the traditional approach. But it also provides superior editing solution, ultimately resulting in enhanced efficiency. And moreover, it facilitates the faster work, ultimately saving the both time and the labor cost. Here is shown a comparison of these two type of sgRNA, chemically synthesized versus IVT. After liposome transfection and electroporation transfection, the chemically synthesized sgRNA show higher editing efficiency, which was consistent with the other reported literature. And chemically synthesized sgRNA cells show lower cytotoxicity and less impact on the host cell line proliferation and cell activity, which was also a consistent with the literature report. And IVT sgRNA involve multi-step biological reaction, and their impurities are like as shown in the blue, is interfere. It's in the during the gene editing or cell engineering work. And while chemically synthesized sgRNA is a stable and has lot to lot stability than IVT sgRNA. So that's ensuring the repeatability and consistency of um, production also. So overall, like IVT sgRNA has the impurity as well. It's degrade after the storage time, while the synthesis sgRNA is remain stable. So a chemical is GenScript is providing synthesis sgRNA. Free online bioinformatic tool for designing the sgRNA and HDR template will help to make appropriate design which can best fit according to the your experiment demand. So for that we have the online available tools where user can enter their target gene, choose an appropriate HDR template, and select the desired sgRNA template, and they can optimize the even sgRNA by adding the different tag for detecting or purifying or localizing the target protein. Additionally, user can specify the quantity, purification method, and delivery format. So that's all the different option available in online tool, and that's make very easy and effective way to place the order for which sgRNA with what modification and what the tag you are looking for can be done. So here shown comparison of different design tools use because this is the source of the GenScript design tools, and A and B is a different design tools which coming from the different vendor. And tool A has the low predicted efficiency, while tool B has the low target efficiency specificity, while GenScript design tool has a more advantage where prediction efficiency with at least higher than the among all the three sgRNA. So specificity score is above 90, and even the predicted efficiency is also very high compared to the another design tool A and B. So one of the sgRNA which GenScript is providing is the Easy Edit sgRNA, and the Easy Edit sgRNA, which support the coming with the superior priority and have the high percentage of full length sgRNA sequence, and which is reliable and consistent for editing results. And it's coming with the different scale also. Along with that, like here I showing the comparing the quality of sgRNA from different vendor. So for that purpose, sgRNA designed for three random sequence 1, sequence 2, and sequence 3, sequence 1, 2, and 3, and it's coming from three different vendor including the GenScript, and all this was analyzed using the UPLC. GenScript Easy Edit sgRNA demonstrated higher purity compared to the vendor 1 and vendor uh, mm, 3. And even in the MS analysis, the sgRNA coming from the GenScript shows the higher percentage of purity. And even we did the full length sgRNA sequence in NGS, and the analysis observed shows that the GenScript shows the consistency even in NGS analysis. And these three different sgRNA means sgRNA coming from three different vendor used for the knockout efficiency study in the Jurkat cell, and GenScript Easy Edit sgRNA shows about the 80% knockout efficiency for all the three gene tested, and sgRNA demonstrated comparable efficiency to other vendor. So another sgRNA which GenScript is providing is a safe edit sgRNA, which is a HPLC purify, has a minimal impact on cell viability and off targeting, and it's ideal for primary and stem cell research and stem cell engineering. So Safe-Edit sgRNA we providing with the default modification for the better stability, and it's coming with the HPLC purified format. So it's giving more than 90% purity, and even the ESIMS guarantee mass variation, which is less than 0.5%, and it can work with the various Cas9 protein. In this figure shows the purity and editing efficiency of safe-edit sgRNA. So safe-edit sgRNA purity for which is greater than 99% for the GenScript, and the competitor product is showing around 42% purity. And the GenScript safe-edit sgRNA shows high percentage editing efficiency also. So we are capable to make high efficient sgRNA with the high purity. So in summary, utilizing the synthetic sgRNA is a simple and cost-effective way to improve your CRISPR editing, as it's offering high editing efficiency, lower cytotoxicity, and higher lot-to-lot stability compared to the IVT sgRNA. At the same time, GenScript sgRNA easy edit and safe edits are very efficient and applicable to the different scale, and shows minimum impact on cell viability. Additionally, GenScript provide add-on QC option like macroplasma testing, endotoxin testing, stabilities testing, and testing. Uh, Even we providing the cytotoxic testing on the client specified cell line. So we also offer to delivery of sgRNA in 96 well plate, which can facilitate high throughput screening also in the early stage discovery work. Uh, so another one which is a homology directed repair means HDR is a natural DNA repair mechanism in cell that use homologous sequence as a template to repair double standard break. While many factors may affect the efficacy of this approach, so choosing the right HDR template is a very essential or maybe critical step. And in the upcoming slide, I will show us the various option of HDR template and their effect. So first of them is gene accept single standard DNA is proven to be one of the best CRISPR HDR template for creating gene knock-in with high editing efficiency and reduce off-target interaction. And GenScript now offer high-quality sequence verified single standard DNA for maximizing the editing efficiency of CRISPR experiments. And the key advantage are highlighted here, which include like lower cytotoxicity. It support the 150 nt to 5 kb insertion available in microgram to milligram scale, which is a sequence verify. At the same time, it's coming with a high purity, and it's produced through our pro priority isothermal production method, and it's enzymatic. It's enzymatic and animal free and easily scalable. And we provide like different scale for this one. So here presented one of the case study where as a single standard DNA HDR template used to produce the T cell engineering. So this work was carried out from Dr. Brian, which from the Marston's lab, UCSF, and their research focus on the large scale editing of primary T cell, aiming at to track locus for the BCMA CAR insertion. And this effort has um, use the HDL template designed by the GenScript, and they did that experiment on the high scale. So here it shows in this figure. They use like clinical stage or clinical scale work and collaborative effort between the GenScript and the Gladstone UCSF Institute resulted in the publication in Nature Biotechnology. So, if you want to know more, you can refer this article, and the publication has detailed methodology for achieving or increasing knock-in efficiency and how the non-viral single standard HDR template help to in the primary immune cell therapy work development. Second is the gene band linear but double standard DNA. Which is but this DNA is covalently close at the end to mitigate the homologous end joining risk and increase the knock-in accuracy. So GenScript universal production vector and thermostable enzymatic process allow for large-scale manufacturing of the long insert sequence. So by using this gene band linear as double standard linear dsDNA, it can support up to 1 to 10 kb insertion, and the key advantage also include like lower toxicity and higher knock-in efficiency compared to the PCR based dsDNA, and the close end which can help to mitigate the non-homologous end connection, and there is absence of unnecessary plasmid sequence, higher stability, so an ideal for long sequence to knock in, and ideal for large scale screening and scale up, and also it's cost effective. And GenScript offer like gram level close ended dsDNA for clinical activity or preclinical activity also. So here I shows like some data for dsDNA versus PCR dsDNA, and we can see like the electrophoric band is a single in case of the higher quality gene band dsDNA, and purity is also shows like about the 99%. And when assessing the knock-in efficiency of the 2.5 kb sequence into the RAB11A sites of HEK293 T cell, we utilize the neotransfection system with the 2 microgram of dsDNA template, and we can see that gene knock-in efficiency of gene band dsDNA higher than that of the PCR dsDNA, because dsDNA gene band dsDNA shows more than 35%, while PCR dsDNA show only 23% knock-in efficiency. So, genuine dsDNA used in engineering TCR T cell. So, this is used by the Neogen Therapeutic, which is a global clinical stage biotechnology company and pioneering in the discovery, development, and manufacturing of next generation engineered T cell receptor TCR therapy. They develop fully individualized neoantigen specific T cell therapy with novel T cell engineering process, and in for that they use the dsDNA. But in order to standardize the TCR gene editing efficiency for clinical application, they decided to source the GMP grade covalently close ended linear double standard DNA template from the GenScript. And when editing the T cell, typically they use 1 microgram of DNA template for the both with and without methotrexate, and editing efficiency reaches up to 85%. However, To reduce the cell viability issue, they tried to reduce the amount of the dsDNA from 1 microgram to the 0.25 microgram. At, and the reduction dose also, the TCR editing efficiency remain about 80%, but the cell viability remains significantly higher. So dsDNA can be work very well into the engineering of T cell therapy, and this is one of the example. The new antigen company used this one. Another one is the case study is from Editas Medicine, is a leading gene editing company which focus on developing the CRISPR medicine for people. CRISPR medicine for people with serious disease. For that, they developed the SLIC technology, and due to the known manufacturing difficulty links to the AAV vector, they investigated whether the slick could facilitate the effective knock-in using the non-viral DNA template in T cell. So they did testing of non-viral DNA template with SLEEK as a potential substitute for the current AAV-based knock-in strategy. For that, they optimized the cell protocol for linear um, mm, uh, dsDNA and ssDNA template. Upon comparing with the sleek um, GFP knock-in efficiency, day 7 viability, and fold expansion among these various DNA template, they observed minimal difference between the ssDNA template and AAV6. So, however, linear double standard dsDNA display slightly slower viability and expansion on day 7 compared to the single standard ssDNA and AAV6. For this experiment, close-ended DNA linear template and single standard DNA template were uh, offered from the GenScript, and it's mm, so this another example how the different template can use for different kind of technology. And another one is the HDR template, which is coming known as a gene circle dsDNA, and which is an innovative small circular double standard DNA vector equipped with very small backbone, and it serves as an effective knock-in template or viral packaging plasmid or non-viral vector. In this vector, both the antibiotic resistance gene and bacterial origin sequence have been eliminated, and hence a smaller vector backbone not only reduce the risk of toxicity and immune response, but they also provide greater stability and boost the transfection efficiency. And moreover, it can support insertion up to 1 to 20 kb, and we can provide them 100 microgram to the gram scale. And moreover, the plasmid will not replicate outside of the engineer production of the host stain. So that's also the advantage of this gene circle dsDNA. And here demonstrated how the gene circle dsDNA improve editing efficiency compared to the standard plasmid. So this is standard plasmid versus gene circle. And we can see the knock-in efficiency increase up to 30% compared to the plasmid in presence of enhancer, and even in non-presence of enhancer also a gene circle dsDNA show higher editing efficiency. So for non-viral T-cell engineering, GenScript offered the Cas9 protein, which we saw a few slides before, and providing the sgRNA, and it's also offered the sequence verified high quality HDR template both into the linear format as well as into the circular format, and to maximize the editing efficiency of CRISPR experiments, and it efficiently support the variety of non-viral based cell engineering application, in including it's maybe used in the gene therapy also. And these are the three different version of the HDR templates available. with three different quality, and the scale also can vary from microgram to the gram scale. So, in summary, as a leader in gene synthesis and genome editing, and through our partnership with Feng Zheng Laboratory at the MIT, GenScript offered the comprehensive CRISPR solution for reliable genome engineering. In addition to sgRNA and Cas9 nuclease and HDR template, we also providing the design tools, even providing the CRISPR gRNA plasmid library, HDR knock-in optimization kit, and CRISPR cell line. So these all different tools can you get from one source and optimize your cell therapy workflow by using this different tool. And the latest FDA guideline advised the use of the GMP reagent before initiating IND studies and clinical trial for the novel cell and gene therapy uh, is necessary. So, to avoid the regulatory hurdle, and researcher must secure the phase appropriate GMP reagent um, mm, with the complete documentation for their IND submission. So GenScript offer necessary expertise and capacity to provide to support the non-viral cell engineering or ex vivo gene therapy. And we provide all CRISPR reagents in the suitable format, like it's coming from REO grade to the GMP like and GMP. So you can select stage appropriate reagent according to which stage of your work you are, and that make easy transition that, and because we are giving the same reagent but the different quality for the different stage, so you're not changing your vendor, but just the quality of material keep getting better and better and coming with the GMP production. So along with the required documentation like required QA and QC, etc. So with 20 years of expertise in nucleic acid synthesis, GenScript has uh, developed very strong capability to establish the um, GMP level CRISPR's reagents. And since 2020, we have assisted numerous clients in the clinical project application. So, like we did more than 1,000 clinical grade batch, sorry, 100. And then even we filed more than 30 IND projects, and seven global IND approval get approved, and we also get 22 successful audits. So all this indicate that we are GenScript is the partner that you can trust if you are developing the CRISPR based cell therapy, and you can rely on us, and you will receive all your GMP CRISPR material on time with supply chain guarantee. And also you will receive your clear documentation and QMS support for IND application, and our professional business development and project management team always available to support your journey throughout to commercialization activity. So, since GenScript has begun to enhance the GMP capability, this involved the not only GMP compliant facility, but we incorporate the GMP management system and even conducted the personal training for that purpose. And as a result, our GMP sites currently operating within the five manufacturing line, and in response to the clinical advancement of our client and the increasing demand for the GMP grade CRISPR material, we have established the new GMP facility, which can equip with the 12 other manufacturing line to accommodate the rising demand. So, and we can provide like 100 milligram to gram with the capacity of 400 batch per year. So additionally we can provide the integrated quality management system. So we provide the comprehensive QA and QC documentation along with the IND filing for different region. So all these are support. GenScript effort is to support the cell therapy or ex vivo gene therapy work to make the customer's life easy and they can get their desired mm, reagents or service on time with the specific quality and with the all kind of supports. So not only that, even GenScript and Avector team up to improve the non-viral cell therapy manufacturing process. So for that, this Avector Solopore platform is automated non-viral delivery system, which offer versatile delivery of different cargoes and gene editing tools such as the mRNA, CRISPRs, and plasmid. So this partnership aim is to advance the cell therapy and make the GMP workflow easy. So you can use the cytosine 1000 equipment for the cell isolation, and then use this platform to make the automated platform to develop the next generation cell therapy into the GMP for the clinical or preclinical work. And in the recent year, so growing number of researchers have been using or exploring the mRNA for development of T cell based therapy. And so for that purpose, here I show like the basic principle of mRNA based CAR T cell therapy. And IVT mRNA leaves no ultimate genetic residue in recipient cell, and it's offer much safer CAR therapy compared to the viral vector based CAR therapy. And mRNA can use for both reprogramming of T cell as well as gene editing with the CRISPR-Cas9 family of nuclease. So manufacturing of mRNA CAR T cell in which the encapsulated mRNA molecule are taken up by the T cell and act as a template for the production of targeting receptor effectively, and reprogramming of this T cell, reprogrammed T cell attack to the cancer cell. So here I presented like how the whole system works. So here the mRNA, and then it's going to T cell and then produce the CAR and then kill the cancer cell. So common mRNA has the five critical structure, a cap, um, then the UTR, and then the therapeutic protein CAR coding sequence, and the polytail length. So cap, UTR, and this is the coding sequence, and UTR and the tail. So crucial factor like plasmid DNA and mRNA integrity and quality of this poly-A tail and proper mRNA capping, even including the lipid nanoparticle packaging, um, all effect into the downstream application and end result. So we, when selecting the mRNA, it's necessary to consider all these different factor like mentioned here, like the key consideration and how GenScript can support this mm, provide the supports and solution. So and here I mentioned what's how its effect. So like if polyethyl quality is not good, its effect on translation efficiency. If capping is not good enough, then its effect on expression efficiency. So one should be very careful while selecting the mRNA and outsourcing from the before outsourcing or taking the mRNA in their workflow. And GenScript provide one-stop shop mRNA from its state-of-art production facility. So here presented data to shows the low integrity impact the expression. So this is the comparison between the GenScript, which has the higher percentage of mRNA purity, and vendor T versus vendor A, which has the lower purity, and how it's affect the expression. So the GenScript showing the higher because the purity is higher, even it's showing the higher expression compared to the vendor T and vendor A. And not only that, but the polyatyl integrity also affect. So that's why we analyze the polyatyl length analysis for all this mRNA coming from different vendor, and we could notice the product from company A has the very flat distribution pattern for polyatyl, and ranging from 30 to 70, and which is usually a characteristic of poly A added by poly A polymerization, while GenScript mRNA show a very sharp peak around 100 to 120, so and shows the uniform distribution and high percentage of population in the desired region. So, Cas9 mRNA is a versatile tool for precision genome editing and has the wide range of application in basic research. So, a comparison of ESP Cas9 mRNA from different vendors was conducted by evaluating the expression of mRNA in A549 cell, and here the data shows that the GenScript shows comparatively higher expression compared to the different vendor. um, and it's available, we have like available and ready to use ESP-Cas9 mRNA in different format. Not only that, like here represent the preliminary data for knockout efficiency in different cell line for different ESP, Cas9, mRNA, sgRNA, LNP composition. And we can see that the two different composition we use, and LNP formulation ALC 0315 shows greater than 80% editing efficiency in both the cell line, like Churkat cell line as well as HEK 293 T cell. So LNP formulation also impacting the editing efficiency. And mRNA synthesis is multi-step process as shown from gene synthesis to the codon optimization, and then cloning and enzyme production and circular mRNA synthesis, and including the lipid nanoparticle formulation. So GenScript is providing the starting from codon to the lipid nanoparticle delivery system, and this can avoid the risk associated, like different material coming from different vendor, and that's make more difficult to get the end product good quality. So GenScript can offer the two-grade mRNA. One is the RU and preclinical. The scale range from zero point 0.2 to 200 milligram, and there are three different capping form available: cap 0, cap 1, cap 2. And even the default length of the polyatyl is 100, but it's also can be customized up to 120. And in addition, GenScript provide the three different nucleotide modification. So moreover, our LNP formulation service also offer four kind of optimized ionizable lipid formulation. And our capability for LNP service can also load RNA in size from 20 NT long to the 10 kb mRNA, and an LNP production can also deliver up from zero point 0.2 milligram to 30 milligram formulation. So all this service also coming from the our Seattle facility, so you can get the made in USA mRNA products for the your cell therapy and ex vivo gene therapy work. Here I highlighted the IVT mRNA QC quality data. So it's showing like our caffeine efficiency is higher. And that's also we carried out the residual DNA, DSNA, and protein residue assay, and which is within the limits. So very good quality mRNA you can get from the Seattle facility. Along with that, we also providing the circular RNA service, and this custom circ RNA synthesis available for different kind of sequence with delivery in only 3 weeks, and the batch scale is vary from 200 microgram to 10 milligram, and it's coming with the HPLC purification and with the guarantee purity is greater than 80%. So along after that, nowadays, new antigen targeted T cell therapy involve the reprogramming of T cell, and it becoming get more and popular. And here I shows the new antigen based immunotherapy diagram. So in that workflow, there are two key steps. One is the identification of new antigen, and second one is synthesize the identified new antigen peptide. For that, GenScript is providing the peptide library, which can help to facilitate the neoantigen identification for the tumors from the tumor cells, or and to meet this, we providing the different kind of peptide library, including like coming crude peptide library or available with the purified peptide library. And we also providing the bioinformatic tools for which can deliver to diverse peptide library as per your requirement. And to support the new antigen peptide synthesis, we also offer GMP new antigen peptide, because our pro-priority neoprem algorithm, which identifies synthetic difficulty, and they can select which synthesis technology used for this new antigen synthesis, and that's where we get the best new antigen peptide, because neoantigen peptide itself has the many challenges while we producing, but the GenScript, we have the expertise and technical capability to synthesize challenging neoantigen peptide in large quantity and provide customized option for the downstream application. And these are the customer testimonial for they using our new antigen peptide. One of them is 3T Bioscience, which using the new antigen peptide from 2017, and they are very happy, and they using them for developing the various kind of T cell based target. And the another one is another is the Avidia technology. They also screen like the different type of new antigen, and for that they making the different kind of new antigen peptide order, and that's make their life more easy to develop different kind of work. So, along with that, we also providing the integrated platform for viral. If you select to go for the viral vector-based cell therapy, we were providing the viral vector packaging service for lentil viral. It's starting from gene synthesis to the lentil virus packaging and coming with all different quality, and we are providing third generation of the lentil virus vector. So this is the research grade, but for we also providing the plasmid, which is a crucial play a very crucial role in the viral vector. So that can serve as a backbone for constructing the viral vector, and for that we providing like basic research grade, early drug discovery, and preclinical grade plasmid, which is coming with different add-on QC option. And due to the time limits, I maybe go very quickly, but if you have any question, you can connect with us. And once you, our GenScript ProBio has the facility to produce the different kind of plasmid into the USA-based facility, and they deliver more than 300 clinical batches and more than 20 CMC project associated with the plasmid production. And the ProBio division also providing the different kind of viral vector, which is very useful for the clinical application and preclinical study also. And the LVV, which is coming both into the adherent and suspension platform, and AV vector also available into the uh, various scale and quality available. But in general, I want to give the one key message that in summary, our cell therapy product and custom solution are designed to meet your clinical manufacturing need which can facilitate the seamless transition from discovery to clinical, and our stage appropriate solution cater to your current and future requirement. So our strength is like we have cutting edge solution for each step of the cell therapy, even including the customization solution available. We provide the quality assurance, and we are provide comprehensive support and committed to oh, support our customer from discovery to commercialization work. So the GenScript Life Science not only providing the solution for cell and gene therapy, but we also providing the complete solution for the vaccine, IVD, cell therapy, gene therapy development. And uh, here I like to conclude my presentation and thank you for joining in today's webinar. I hope you found it informative and insightful. So, due to the time limit, I maybe last few slide I go very quickly. But if you have any question, please feel free and connect with us or send us the email, and we will definitely help you to your work. Thank you.